carbon dioxide joins with 5-carbon ribulose biphosphate. Now this is part of the light-independent reactions, and this occurs in the stroma of the chloroplast. The carbon dioxide is being reduced in this process, and it produces 2 3-carbon glycerate phosphates, known as GP. The next step after that is the consumption of the products of the light-dependent reactions, and that produces triose phosphate. Philosophically, the way to look at this is it produces reduced carbon, so carbon-hydrogen bonds. And those reduced carbon molecules are used to then make everything in all living organisms. So it's made into amino acids, it's made into fats, it's made into carbohydrates, it's made into nucleic acids, and it's made into everything that makes you, because everything in you was previously in a plant or in an animal that ate a plant. Now, how this process is powered and where the electrons come from to reduce the carbon is that we have 2 molecules which transfer firstly energy and secondly electrons. So we need an input of ATP, and the ATP is hydrolyzed and becomes ADP. We also need an input of electrons. And these electrons are carried by the carrier molecule NADP, and when it's reduced it's NADPH, and it delivers those electrons and becomes NADP. In order for the light-independent reactions known as the Krebs cycle to proceed, you need carbon dioxide, you need ribulose biphosphate, you need ATP, and you need NADPH. So you need a source of energy and a source of electrons. And that's going to enable you to reduce carbon. Now, where that comes from, where the ATP comes from and where the NADPH comes from, is it comes from the capturing of light by the light-dependent reactions. So this requires some membranes, and along those membranes flow electrons. And as those electrons flow along the membranes from photosystem 1 to photosystem 2 to photosystem 1, the electrons flow along the membranes, and they're pushed by the energy from chlorophyll. As they do that, they push hydrogen ions across the membrane, and those hydrogen ions flow out across that membrane, and that drives the phosphorylation of ADP to ATP. Those electrons, which have been pushed from photosystem 2 to photosystem 1, then get picked up by NADP, forming NADPH. So it's the electrons that have been lost from photosystem 2 that get pushed along from photosystem 2 to photosystem 1, and these electrons finally end up reducing NADP to NADPH. Now this leaves us a problem in that photosystem 2 is now missing a couple of electrons. So what we have, the process here, is we have a process called photolysis. And lysis means to split, and photo is with light. So in this case we take water and we split it to give us the electrons, some hydrogen ions, and those hydrogen ions help to contribute to the gradient of hydrogen ions that flow out through ATP synthetase, generating ATP, and also it produces some oxygen. We'll put the oxygen coming off at the bottom here. So what we have is a process that uses enzymes, occurs in the stroma of the chloroplast, and is a reductive process which is taking carbon dioxide and reducing it to produce these carbon-hydrogen bonds which are used to make everything in all living organisms. And this is autotrophic. We power that by a process of light capturing, where we split water by capturing the light in photosystems and moving the electrons along an electron transport chain on a membrane. That movement of electrons pushes hydrogen ions from where there's few of them to where there's lots of them, which is inside the thylakoid membrane. They then flow out through ATP synthetase, generating ATP, and they then finally join with NADP, forming NADPH. The NADPH is then used to carry on the Krebs cycle and to regenerate the IUBP. So now what we're going to look at is how that connects to aerobic respiration. So if we consider aerobic respiration occurring after glycolysis, so we're looking here at the link reaction and the Krebs cycle which occurs in the matrix of the mitochondria. If we imagine that this reduced carbon, this has been turned into glucose and carried out glycolysis, so we start with our 3-carbon pyruvate. Now that 3-carbon pyruvate, remember, gets decarboxylated to produce carbon dioxide and dehydrogenated to produce reduced NAD. That gives you a 2-carbon fragment which joins with a 4-carbon, and that gets broken down, 6-carbon to 5-carbon to 4-carbon and back to 4-carbon, and this is the Krebs cycle or TCA or citric acid cycle. Each time there's decarboxylation, there's removal of electrons which are used to reduce the NAD, and there's another one here. And there's another one here, and there's an FAD, we're going to ignore that for a minute. Also, we've produced a carbon dioxide here, we're going to lose a carbon dioxide here, and a carbon dioxide here. So these are our 3 carbons in the pyruvate has been decarboxylated, decarboxylated, and decarboxylated. The reduced coenzymes, so the NADH, what happens to these things is that they go off to a layer of membranes, and the layer of membranes are the inner mitochondrial membrane, and what happens there we've got an electron transport chain, and this NADH delivers the electron and that regenerates the NAD. That electron then goes down the chain of carriers, and there's a series of oxidation and reduction reactions where the energy in that electron is used to push protons from where there are few to where there are many across the membrane. And remember, this is the inner mitochondrial membrane and this is the outer membrane. At the end of this process, this is where oxygen is used. So this oxygen here that's being produced by photolysis is used to join with the electron and a hydrogen to produce water. The the hydrogens that you've pushed from the matrix into the intermembrane space, they then flow out through ATP synthetase, and they generate, as they flow out, they generate ATP. Now, the carbon dioxide that's produced in the link reaction and in Krebs cycle, well, that is the carbon dioxide that's used here in the Calvin cycle. So philosophically, we've got a process that takes light energy and uses it to split water to produce the electrons which are used to reduce the NADP to NADPH. Those are then used to reduce the carbon dioxide. ATP is produced by moving electrons along a chain of carriers and using that to push protons across the membrane, which flow out producing ATP through ATP synthetase. Oxygen is produced as a waste product. But in respiration, we take the reduced carbon that we've produced in the Calvin cycle, and that's decarboxylated. Now remember, the oxygen here with the carbon dioxide has absolutely nothing to do with the oxygen that you breathe in aerobic respiration. As we decarboxylate it, we dehydrogenate it, and we use the electrons to reduce the NAD. The NAD produced from the link reaction and from Krebs cycle goes off to the inner mitochondrial membrane, where it delivers an electron and goes from being reduced NAD to being oxidized NAD. That electron travels down a chain of carriers in the inner mitochondrial membrane. As it does that, it pushes protons across the membrane, just like in photophosphorylation. And at the end of it, the electron on the last carrier, that electron is removed and the carrier is oxidized by taking the electron and joining it with oxygen to produce water. And the oxygen that you're joining it with is the oxygen that's being produced here by photolysis. And that the carbon dioxide that you produce in Krebs cycle is the carbon dioxide that gets consumed in the Calvin cycle. Okay, that's longer than normal, but I hope that makes some sense.